everyone, I am Dr. Preeti Khulbe and my today's topic is pilot plant scale-up technique. So in previous videos we have discussed about the many aspects related to the pilot plant studies. So if you want to know all these aspects, that is the introduction, the general consideration related to pilot plant, what is the difference between pilot plant and scale-up, what is the need of scale-up, advantages, disadvantages, GMP consideration, that is good manufacturing practices consideration related to pilot plant studies, you can visit my previous videos. Also, I have made the videos related to the pilot plant scale up of different dosage forms, like we have discussed about the solid dosage form pilot plant, we have discussed about the pilot plant of liquid and many more. So if you want that detail, so you can visit my previous videos. This is the 6th video of this pilot plant scale up series. So first of all, learning objective. So learning objective related to this topic is the pilot plant, to know the various things related to the introduction and methods of scale up, why scale up is important and what are those methods by which we can achieve the scale up. Okay, this is the major thing which we will came to know by using after the studying of all these slides. Other than that, know the various aspects related to the pilot plant, that is what are the general requirement, what are the space requirement, what are the general things related to the pilot plant, what we need, what are the things which we required for the pilot plant and the scale up. This will all we will discuss in our in this topic. Other than that, GMP consideration related to the scale up, that is why good manufacturing practices is essential, and why we are studying this, and why it is related to the scale up. So all these things we will discuss in this topic. Other than that, learning outcome of this pilot plant scale up unit is need of scale up, why scale up is necessary, pilot plant of various dosage form. As I discussed that we have already discussed about the pilot plant of solid dosage form, the tablet, and the liquid dosage form, suspension, emulsion, solution. All these things we have discussed. We have also discussed the pilot plant of coating thing. So you will came to know about all these things. So you can if you want to know all these about all these things, so you can visit my previous videos. Now, advantages and disadvantages of scale up techniques also you will came to know after the completion of this complete unit. Now the thing is scale up of semi-solid dosage form. So our today's topic is scale up of semi-solid dosage form. As we have discussed about the solid dosage form, that is tablet, we have discussed about the liquid dose form scale up, that is solution, suspension, emulsion. So I think it is very easy to to know about the scale up of semi-solid dosage form, as this type of dosage form is it is in between the consistency of solid and liquid. And you know that when we are studying about the solid and liquid dosage form scale up consideration, so we have discussed that we have to find out the steps involved in that particular manufacturing process of that particular dosage form, and after that we have to pick the we can say we have to pick the critical steps of that particular process. Okay, so that means we can say that in this scale up of semi-solid dosage form also, we first need to understand what is the process, why the process is important, what are the different steps which are involved in the process, and then we can easily easily find out the critical parameters, and we can scale up on the basis of that parameters. Okay. Now, semi-solid dosage form in journal. What is semi-solid dosage form? So, in journal, semi-solid dosage forms are complex formulations having complex structural elements. Often, they are composed of two phases, that is oil and water, one of which is a continuous, that is the external phase, and another one is the dispersion or dispersed phase. So, semi-solid dosage forms, means for one phase should be should be the solid in this case, and other phase may be oil or maybe water. The active ingredient is often dissolved in one phase, and although occasionally the drug is not fully soluble in the system and is dispersed in one or both phases, thus creating a three-phase system. Okay, so it can create it, the three-phase system also can be created by dissolving the active ingredient in one phase, and then make a dispersion with the one or both phases. Okay. Now, semi-solid dosage forms, the physical properties of the dosage form depends upon various factors. Okay, so this dosage form, actually physically these dosage forms are different. So the physical property of this type of dosage form depends upon various factors, including the size of the dispersed particles, the interfacial tension between the phases, the partition coefficient of the active ingredient between the phases, and the product rheology. So all these things, the physical property may change. If the physical property may change, so it may change the interfacial tension, it may change the dispersion particles, and it may also change the rheology of the product. That is, rheological property of liquid is different and semi-solid is different. So these factors combine to determine the release characteristics of the drug, as well as other characteristics such as viscosity. So obviously you know that the semi-solid formulations are viscous formulations. So the release characteristic from the dosage form of semi-solid dosage form is dependent upon the viscosity of that particular formulation. Now the next is suppositories. So the manufacturing of suppositories on a laboratory scale usually involves, so the one thing which is a type of semi-solid dosage form is suppository type. So in case of suppository, there are multiple steps which are there in the laboratory preparation of suppositories. So in this, first one is the preparation of a molten mass. So that is, first we need to prepare first we need to prepare the mass. Means we need to we can say melt the base which we are taking. Then the dispersion of drug in the molten base. Then we have to disperse our base, which disperse our drug, the active ingredient or the API. We have to dissolve, we have to disperse basically on the melted base. Okay, that is, for example, we are taking beeswax as a base. So first we need to melt it completely, and after melting completely, we have to disperse the drug into it. Okay, so yeah, so this is that. Then casting of suppositories in a suitable mold. We have to take the suppository in the mold, and we have to we can cast it. Then cooling of the mold. After casting, we have to cool the mold in the lower temperature. For the for the melting of the base, it is essential to increase the temperature, and for the cooling of the mold, obviously we need to decrease in temperature. So here you can see that there is a temperature fluctuation in the process. So we can say that if the drug is having the instability in temperature fluctuation, so it is not suitable for the suppository. So cooling the mold. After cooling the mold, we have to open and remove the suppository. Okay, when we cool the mold, so mold will ultimately leave the suppository. That means no sticking of the mold and suppository is there. It will leave easily, and we can easily collect it, open it, and remove the suppository. More number of molds and large size pan for melting of drug and bases. So if we need a large size, if we need to make a large base system or large size system of suppository, then we need number of moles. We can increase the number of moles, and we can increase the size of the pan. That is, we can take the large size of the pan for this process. So these are the steps of making the suppository. First we need to melt the base which we are taking, then we disperse the drug into the melted base. Then after making this dispersion, we have to cast the suppository into the molds, any type of, any shape, any type, any shape we can take for the for making the suppository. Which type of mold shape we are selecting, it all depends upon the the type of formulation we need. Okay, yeah. So next is we have to then cool the mold, so that it can easily it can easily leave the suppository from the wall. Then open and close the suppository. So that means we have to we can open and we can remove the suppository easily from the mold. Okay, and if we want the large size, so we can go for the number of molds we can increase, and we can increase the large size of pan. Now the parameters. So for a true solution, order in which the solutes are added to the solution is usually unimportant. The same can't be said for dispersed formulations. However, because dispersed matter can distribute differently depending on to which phase a particular substance is added. Okay, so the phase of the substance or addition of the substance is very, very important, right? In a typical manufacturing process, the critical points are generally the initial separation of a one-phase system into two-phase system. So here this is very important that we need to check the either the system is breaking or not. Okay, so that means two-phase system you can take, and you can convert one-phase system can be converted into two-phase. So that will be your critical point. So that means you need to understand that this critical point you have to take into consideration while you are designing the formulation for the scale-up, and the point at which the active ingredient is added. So another important thing which is related to this is one phase system to this which is related to this is while we are adding the active ingredient. So at what time we are adding the active ingredient, and if there is a fast phase separation or not. So this you have to consider while you are designing a two-phase system, or you can say a suppository system. Now, because the stability of each added ingredients is important for determining whether a mixture is visually a single homogeneous phase, such as data possibly supported by optical microscopy should usually be available for review. Okay, so because the solubility of each ingredient is different, so it is very important to determine the all the solubility related things to each and every ingredient. And mixture is usually a single a single homogeneous phase, such as data possibly supported by optical microscopy, and should usually be available for the review. Okay, so all the things which are related to this are is under the review, and it is easily available for it. Okay, so this is particularly important for solutes added to the formulation that that at a concentration near and exceeding that of their solubility at any temperature at to which the product may be exposed. So it is very essential to know the solubility of any product which in any temperature, and this is particularly very important for the solute, that if we are adding such a solute in any concentration which are near to its solubility at any temperature, which may exposed to the product. Next is variation in the manufacturing procedure that occurs after either of these events are likely to be critical to the characteristics of the finished product. Okay, so if we want to check the variation in the manufacturing procedures, so it also may be added as a critical ingredient for the finished product. This is especially true of any process intended to increase the degree of dispersion through reducing droplet or particle size, that is homogenization. So for any process, the homogenization is also, we need to study related to it. We need to focus on the homogenization of the product. Then aging of the finished bulk formulation prior to packaging is critical and should be especially addressed in process validation studies. So that is aging of the finished bulk formulation is basically how much time we have stored the formulation before packaging. Okay, for example, we don't have packaging material at that time, at the time when we are we are making the formulation. So how much time we have taken it into the hold before packaging, हमने कितने टाइम के लिए प्रोडक्ट को स्टोर किया है, that also we need to clearly mention. We need to work, we need to review in the process validation study. Now, journal stability consideration. So the effect that SUPAC changes may have on the stability of the drug product should be evaluated. For journal guidance on conducting stability studies, see the FDA guideline for submitting documentation for the stability of human drugs and biology. So FDA guideline is there, SUPAC guidelines are there, so for the stability study, so you can check it. Now, journal stability consideration for SUPAC, the submissions, the following points should also be considered. First one is in most cases except those involving scale up, stability data from pilot scale batches will be accepted to support the proposed changes. Where stability data show a trend towards potency loss or decredent increase under accelerated conditions, it is recommended that historical accelerated stability data from a representative purchases batches be submitted for comparison. Okay, so it is very essential that we need to check the stability study, and as during the SUPAC submission so we need to submit the all the stability data for the batches, and we can do the stability accelerated stability studies, and we can submit the long-term data which is representative to the previous batches. So now as we are discussing about this SUPAC guidelines, so what is SUPAC? SUPAC is scale up and post approval changes. So if there is any post approval changes and scale up, so you have during scale up, if you are doing certain changes and post approval changes are there, so you have to submit the document, SUPAC document. So this is what the SUPAC is. Now it is also recommended that under these circumstances, all available long-term data on test batches from ongoing studies be provided in the supplement. So long-term data which are having which is of the previous batches, we need to submit along with this, okay, as a supplement. Now the submission of historical accelerated and available long-term data would facilitate review and approval of the supplement. Now, journal stability consideration. A commitment should be included to conduct long-term stability studies through the expiration dating period according to the approved protocol on either the first or first three production batches. Okay, for this section 3 to section 6 are there, and to report the results in subsequent annual reports. Okay, so journal stability consideration mein we can say that for the commitment, it should be included to conduct the long-term stability studies. So it is there that long-term stability studies is performed, and the its expiration dating and all we have to mention, and according to the approved protocol on either the first three batches, we need to take it into the consideration, and we need to submit it along with the annual report. So that is if it is our first batch, so not only one batch data, we have to present the 3 batches data, and first 3 batches should be there, and it should be continuous batches, continuous first 3 batches should be there. Now the testing. The key parameters for any drug product is its efficiency and demonstrated in controlled clinical trials. The time and expenses associated with such trials make them unsuitable as routine quality control methods. Therefore, in vitro surrogate tests are often used to ensure that product quality and performance are maintained over time and in the presence of the changes. So the time and expenses associated with such trials make them unsuitable as routine quality control methods, and that's why the in vitro surrogate tests are often used for the assurance of the product quality and its performance over the period of time, or maintained time, we can say, in the presence of any changes. If there is a a change in any in vitro performance, so we can consider it. Now, variety of physical and chemical tests commonly performed on semi-solid products and their components. So, when we talk about the semi-solid dose form, as we have discussed in the tablet also, in the liquid dose form also, there are certain tests we should perform to know the quality of that particular product. So, to know the the component, the physical and chemical test performance of that to know the performance of that semi-solid products, we have certain tests which we can use for the semi-solid dosage form. So these tests are solubility, particle size and crystalline form of the active component, viscosity and homogeneity of the product have historically provided reasonable evidence of consistent performance. So here you can see that multiple tests are given. Solubility is obviously essential to know, because if the product is not properly soluble, then it will not give proper dissolution. So solubility is the first thing which we should check for the semi-solid product. Then particle size, we have studied about particle size a lot, that particle size is essential for the effective release of drug. Obviously, if the particle size is not proper, the solubility is not proper, and then drug release is not proper, the crystallinity or crystalline form of drug will also dependent upon the many characteristics. Like if the crystalline form is there, their solubility is different, their pattern of release is totally different. So we need to understand this also. We need to check the viscosity, we need to check the homogeneity of the product. Now, more recently, in vitro release testing has shown promise as a means to comprehensively assure consistent delivery of the active components from semi-solid products. So in vitro release testing is very essential for any type of formulation, because so that we can compare it and we can comprehensively ensure the consistent release or consistent delivery of active ingredient, because we cannot perform the study directly to the in vivo, and in vivo the pattern we cannot know before doing the in vitro study. So in vitro study previously we need to do, then we can changes, then we can compare it for the in vivo studies. Okay, so that's why the study of solubility, particle size, crystalline form, active component, viscosity, homogeneity, and the in vitro release is important for the testing of semi-solid drugs. Now the role of in vitro release. So an in vitro release can reflect the combined effect of several physical chemical parameters, including solubility and particle size of the active ingredient and rheological properties of the dosage forms. In most cases, in vitro release rate is a useful test to assess the product seamless between the the pre-change and post-change product. So that's why it shows, we can say it shows how the drug is releases, how the active ingredient is releases, how the rheological properties influences the dosage form, etc. Now the role of in vitro release. However, there may be instances where it is not suitable for this purpose. In such cases, other physical and chemical tests to be used as measured of sameness should be proposed and discussed with agency, whatever the agency is you are enrolling for. With any test, the metrics and statistical approaches to documentation of sameness in the quality attributes should be considered. The role of in vitro release. The evidence available at this time for the in vitro in vivo correlation of release test for semi-solid dosage forms is not as convincing as that for in vitro dissolution as a surrogate for in vivo bioavailability of solid oral dosage forms. Therefore, the center's current position concentrating in vitro release testing is as follows. So the center's concurrent position concentrating in vitro testing as follows. Number one is the in vitro release testing is a useful test to assess product sameness under certain scale-up and post-approval changes for semi-solid products. So in vitro release testing, we can say, is a assessment of sameness. That is, the product, the post-approval is same and pre-approval is same if there is any changes. So release characteristic we should submit in the SUPAC also, and in any case of things if we apply it for, in any case of agency we are talking about. Okay, so the development and validation of an in vitro release test are not required for the approval of an NDA, ANDA and AADA, nor is the in vitro release test required as a routine batch-to-batch quality control test. So if we are producing a new product and we need anything for this for the approval, any approval we want, either we want NDA, new drug application, ANDA, abbreviated new drug application, abbreviated drug application, abbreviated drug application. So any type of thing if we want, then we need to submit the data related to the in vitro study. So it is very essential. Now third point is, in vitro release testing alone is not a surrogate test for in vivo bioavailability or bioequivalence. So if we want to check, if we want to know the bioavailability and bioequivalence study of certain things, so in vitro release testing plays a very important role for this. The in vitro release rate should not be used for comparing different formulation across the manufacturers. So These are the references. So in today's lecture, we have discussed about the scale-up consideration of semi-solid dosage form, in which we have discussed about what is semi-solid dosage form, what do you know about how we can prepare the semi-solid dosage form, then what are the properties, which are the essential factors which we need to consider when we are designing a semi-solid dosage form. Then we have taken an example of suppository. So we have discussed about the steps involved in the suppository. Then we discussed that what are the parameters should be there while we are designing semi-solid dosage form. Now we have, along with this, we have also discussed about that there are certain critical parameters which we need to consider when we are doing the scale-up technique. Then we have discussed about the general stability consideration related to this technique. In this, we have discussed about the SUPAC changes. SUPAC is scale up and post approval changes, and we also have discussed about the FDA guideline related to this. So we have discussed about general guidelines, general stability consideration which we need to submit in the SUPAC. Other than that, we have discussed about the general stability consideration, testing, and the role of in vitro release testing in different fields. Okay, so these are the things we have discussed in today's lecture. So references, I can say that the first you need to go for the books. There are many books. There are each and every pharmaceutics book in which the scale-up, pilot plant scale-up topic is given. So first you need to understand what is scale-up, what is the general consideration related to scale-up from the books. Then you have to go for the journals for the latest update related to the scale-up on pilot plant. Okay. Now come to the next, that is the the objective questions. So first objective question is, control factor of semi-solid in pilot plant does not include. Okay. So you need to understand the answer now. The control factor which are not there in the semi-solid. Speed of mixing, obviously because mixing is there, so speed of mixing we have discussed, so it is an essential factor. Rate of material feed, always rate of material feed is the critical factor. Type of equipment is also there, which is also a critical factor, so that means answer D, that is all mentioned, is the we can say the right answer for this. Now the second question is, critical factors of liquid in pilot plant not include. Now in previous video we have discussed about the liquid dosage form scale up. So now you need to understand what is the critical factors of liquid is not included. It is mixing tank size, yes, it is important. Mixing time is important, mixing rate is important, material type is not so important in this case, so that means the answer is D, material type. Next is, what is SUPAC? So we have discussed a lot about SUPAC here. So SUPAC is scale up and pre-approval changes. SUPAC is safety and precautionary approval changes. SUPAC is scale up and post-approval changes, or SUPAC is safety and pre-approval changes. So the right answer is scale up and post-approval changes. Next question is, which is true about SUPAC? So we have discussed that SUPAC is scale up and post approval changes. So now what is the true sentence which is related to the SUPAC? Only accelerated stability data are required in SUPAC. B. Only long term stability data are required. C. Only the annual report is required. Or D. Data of accelerated, long-term and annual reports are required. Okay, so when we talk about the SUPAC, so we should be clear that we need to submit all the data. It is data related to accelerated study, data related to long-term and annual reports. All the data are required. Next is, the next question is last step of compression cycle is, actually in this video I have mixed some questions related to previous videos also, so that it is easy to remind you the things of the previous video. So last step of compression cycle is, the answer is ejection. Filling of dye is not there, pre-compression is not there, and compression is not there. So that means ejection is the last step of the compression cycle. Next is reduced free flow of granules is due to overloading, not proper mixing, underloading, or more mixing time. So answer A is correct, that is overloading. Next is, which is not important in scale up or dry granulation? Forced used in slugging, diameter of punches, mixing speed, and sizing. So C is the correct answer, that is mixing speed. The next thing related to this is scale up of blending in pilot plant includes, first one is blender size, second is mixing time, third is mixing speed, and fourth one is all mentioned. So as we have discussed in the blender or in any type of equipment, all the things are important. So size is important, time is important, and speed is important. All the things are important. So that means the answer is all mentioned is correct. Next question is, control factor of milling in pilot plant does not include. So milling की यहाँ पे बात हो रही है, so you should focus that milling control factor does not include speed of mill, date of material feed, type of equipment, and brand of equipment. So obviously speed is important, date of material feed is important, type of equipment is important, but brand of equipment is not so important. So answer D is the correct one. Yeah. So that's all for today. So in this lecture, in today's video, we have discussed about the scale up of semi-solid dosage form, and in this we have discussed about the general consideration related to scale up of semi-solid dosage form, the how we can prepare the semi-solid dosage form, and what are the specific steps in the suppository making, how we can test the how we can do the testing related to these things. And also we have discussed about some SUPAC guidelines related to this, related to the testing and stability. We have discussed about all the stability testing, why it is important, or what are the need of doing this stability studies and other related testing. We have discussed that solubility testing is essential, the viscosity testing is essential, particle size is essential, so in vitro studies obviously essential. So all these things we have discussed in today's lecture. Other than that, we have also discussed about some some references related to this. You can also not only these references are important, you can also go for the more references if you want. Then objective questions we have discussed. We have discussed the objective question related to the today's video as well as the previous video for the revision. So that's all for today. Thanks for watching my video.